Hello everyone. In today's video we are going to learn what are intercalating agents and their mechanism. Intercalating agents are flat molecules which can slip between bases in a DNA helix that can slightly unwind the DNA helix and increase the distance between base pairs. Suppose these are base pairs on a DNA helix. This is an intercalating agent that can enter between two base pairs and increase the distance between these bases to cause mutation in a DNA. Now, there are two types of mutation that can be caused by an intercalating agent. They are Frameshift mutation by addition and Frameshift mutation by deletion. For those who are unfamiliar with the term Frameshift mutation, it is a kind of mutation that is caused by entering or deleting one base pair inside of a DNA sequence to completely change the DNA sequence. For example, there was some bases like this A, G, C, T. Now, if one base enters here, that changes this G's position from 2 to 3. This was in the second position before, but when something enters in between this becomes the third base pair, and this causes the whole sequence behind this to change their position. Now, Frameshift mutation by addition is the similar process. Here is the type 1, that is Frameshift mutation by addition. The Frameshift mutation by addition is caused when the replication of DNA occurs. Here we have a template strand and a new strand that we are familiar with in a replication. When a replication occurs, if this is an intercalating agent that enters in between two bases and causes a distance to occur here, as it is the new strand and this doesn't have any opposite base pair here, so this takes a random base inside it. For example, we can say a G. Now we can see that the template strand had only 10 bases, whereas the new strand has now 11 bases, including this random base that entered here because there was no other complementary base in the template strand, but there is a distance. So to fill this distance, the DNA just chose a random base in this place. Now when this new strand goes into replication, this becomes now the template strand, and when it is replicated, so in this step we saw this G entered between C and A. So when it is a template strand, this becomes the new template strand. Now when replication occurs, this strand will have the opposite bases of this template strand. So in opposite of this G, it will have a C. Now see that new replicated strand should have the same base sequence like this one, but here as the intercalating agent entered here, the subsequent new replicated strand became 11 bases, and the sixth base is C, the rest of the bases has changed the positions. Here the TT was on 6 and 7, but here they are 7 and 8. Like this, the whole next part of the strand changed their position by one number. That was 6, 7, 8, 9, 10, now have become 7, 8, 9, 10 and 11. This is Frameshift mutation by addition. Now for the type 2, that is Frameshift mutation by deletion. Here we have taken the template strand as same of the previous example. Here we have 10 base pairs of a template strand and a new strand that is TAGTC AATGA. Now, in case of frameshift mutation by deletion, an intercalating agent replaces a base. That, for example, this C has been replaced by an intercalating agent. So, the base pair, the base sequence has become TAGT AATGA. But when this goes into replication, this intercalating agent doesn't stick to the DNA sequence and gets rid of here. So the C has been replaced, but nothing has taken its place. So the new template strand has only 9 bases, that is missing the C. But as the intercalating agent has been removed, so there is nothing that can replace it here. So the replication continues with that 9 base pair. As a result, the new replicated strand becomes its complementary, that is ATCA TTACT. Now we can see in the first template strand there was 10 base pairs where the TTACT was on 6, 7, 8, 9 and 10th position. But here, as the C of this was replaced by an intercalating agent, the new template strand became a 9 base strand. As a result, the new replicated strand has 9 bases and is missing the G because there was no C here. So the TTACT has become now 5, 6, 7, 8 and 9th base in place of 6, 7, 8, 9 and 10th base. This is another frameshift mutation that is caused by deletion. This is how intercalating agent causes frameshift mutation by addition and frameshift mutation by deletion.